Am I not funny? Am I not funny? Tell me I'm not funny, man. I mean, look at this. What if I just... Oh. 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 This is kind of funny, guys. Like, look at me. I look really short, right? Hey! Hey! Open the door! Open the door! Let me in! Or I can be really tall. I think this is like height accurate. Yahoo! 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 I'm here! I'm alive! We're not gonna be reading yaoi today, unfortunately. I'm trying to get used to the content creator shit and clickbait or whatever. Can you hear this? Ah. Apple. Apple. A-P-P-L-E. Apple. What should I do for the camera view? Would it be funnier if I just like... I'm gonna just do this the whole time. If it comes to this, like, what the fuck, man? What are you doing, Usa? That's pretty awesome. I think I'm pretty awesome. Wow. What was I gonna do today? Oh yeah. We're not reading yaoi. We're reading something you could debatably say is better than yaoi. And that is biology. Reading 300 pages of a biology research book or whatever. I feel like I'm gonna regret this, low-key. I might actually. Today I'm using Spout. I saw a tweet today that was like, guys, what is Spout? And why are you capturing that instead of YouTube Studio? And I was like, what do you mean? Like, I was like, yeah, like, what is Spout? What the heck is Spout? And then turns out I've been abusing my CPU this whole time because I've just been directly capturing Spout. Or not directly capturing Spout, but directly capturing OBS. It's terrible. It's freaking terrible. Alright, I'm a little bit scared. I have my windows open right now. And it's like pitch black outside. I just wonder if there's like somebody watching me through my window. Should I close my window? Alright, I'm gonna close my window. I'm kinda scared. Okay. Close my window. You're like, hi my future VOD watchers. You're probably gonna see this and you're gonna be like, what the fuck are you doing, Usa? Sorry. That would be really boring if you had to stare at my face, my usual face. As always. So, yeah. You'd have this instead. Police, that's her. Moshi moshi, 警察ですか? Yeah. 私? It's not me! Leave me be! Ah. Oh wait, this angle is so bad. Holy, wait. Okay. Cause if I go down here, it's like my head's so. I'm like Mega Mind. If I go here, that's not more accurate though. I'll go like here. So, どうですか? Kawaii, kawaii no. As I was saying though, I was like, what is it? The my VOD watchers, you guys are probably gonna see this here and be like, what the fuck are you doing, Usa? And it's just vibes, man. This is just vibes. Like, you should be thanking me. I'm like keeping you less bored by keeping my camera angle like this. Okay. I'll start reading. Guys, what if this VOD is like 8 hours long and then you're not gonna sit through and watch the whole thing because it's gonna be freaking 8 hours long, man. I don't wanna do this. Boo. Boo. This sucks. Booty cheeks, man. Just kidding, this is adulting. This is adulting, right? This is adulting. Chapter 1. The Cell. Microfilaments. Microfilaments are made up of solid polymerized rods of actin. The actin filaments are organized into bundles and networks and are resistant to both compression and fracture, providing protection for the cell. Actin filaments can also use ATP to generate force for movement by interacting with myosin, such as in muscle contraction. Microfilaments also play a role in cytokinesis, or the division of materials between daughter cells. During mitosis, the cleavage furrow is formed from microfilaments, which organize as a ring at the site of division between the two new daughter cells. As the actin filaments within this ring contract, the ring becomes smaller, eventually pinching off the connection between the two daughter cells. Did any of that stick? Because I know for me it didn't. I need to check my audio. Okay, Usa, speak for me. Damn, my delay's kinda ass, man. What the hell? Wait, you're kinda crazy. What the fuck, Usa? It's okay. It's okay. It's cope. This is cope. Continuing on. Microtubules. Tubules. Unlike microfilaments, microtubules are hollow polymers of tubulin proteins. Microtubules radiate throughout the cell, providing the primary pathways along which motor proteins like kinesin and thionine carry vesicles. Cilia and flagella are motile structures composed of microtubules. Cilia are projections from a cell that are primarily involved in the movement of materials along the surface of the cell. For example, cilia line the respiratory tract and are involved in the movement of mucus. I'm actually gonna fucking cry. 恥ずかしい。マジで恥ずかしい。POV, some random 5 foot chick standing outside your door reading you some cryptic research paper. Where was I? For example, cilia line the respiratory tract and are involved in the movement of mucus. Flagella are structures involved in the movement of the cell itself, such as the movement of sperm cells through the reproductive tract. Cilia and flagella share the same structure, composed of 9 pairs of microtubules forming an outer ring, with 2 microtubules in the center. This is known as a 9+2 structure and is seen only in eukaryotic organelles of motility. Bacterioflagella have a different structure with a different chemical composition, as discussed later in this chapter. Motor proteins like kinesin and dynein are classic examples of non-enzymatic protein function, along with binding proteins, cell adhesion molecules, immunoglobulins, and ion channels. Motor proteins often travel alongside cytoskeletal structures to accomplish their functions. Non-enzymatic protein functions are discussed in There's more ASMR. Plasma membrane. Repeat after me. Plasma membrane. Dynein arms. Dianin. I can't say it. Blah. Blah. Dynein arms. Dynein arms. Plasma membrane. Why aren't you asleep? I thought I killed you, man. You should have died. In Minecraft. It sounds really bad. Like out of context. Central microtubules. Radial spokes. Radial spokes. Nexon. He hates me? I'm saving you, man. I'm being so generous. Inner sheath. B tubule. A tubule. Doublet microtubule. Who the fuck is naming this shit, man? Hey! No swearing. Centrioles found in a region of the cell called the centrosome. They are the organizing centers for microtubules and are structured as 9 triplets of microtubules with a hollow center. During mitosis, the centrioles migrate to opposite poles of the dividing cell and organize the mitotic spindle. The microtubules emanating from the centrioles why did I read it like that? The microtubules emanating from the centriole attach to the chromosomes via complexes called kinetochores and exert force on the sister chromatids, pulling them apart. None of this is sticking, man. None of this. Intermediate filaments are a diverse group of filamentous proteins, which involve keratin, desmin, thymidin, and lamins. Many intermediate filaments are involved in cell adhesion or maintenance of the overall integrity of the cytoskeleton. Intermediate filaments are able to withstand a tremendous amount of tension, increasing the structural rigidity of the cell. In addition, intermediate filaments help anchor other organelles, including the nucleus. The identity of the intermediate filament proteins within a cell is specific to the cell and tissue type. Words be wording for real. Dude, it doesn't even look like English anymore. It feels like I'm reading hieroglyphics or something. Okay. One of the unique characteristics of eukaryotic cells is the formation of tissues with division of labor, as different cells in a tissue may carry out different functions. For example, in the heart, some cells participate in the conduction pathways while others cause contraction. Still, others serve a supportive role like maintaining structural integrity of the organ. There are 4 tissue types: epithelial tissue, connective tissue, muscle tissue, nervous tissue. While muscle and nervous tissue are considered more extensively in subsequent chapters, we explore epithelial and connective tissues below. Epithelial tissue. Epithelial tissue covers the body and lines its cavities, providing a means for protection against pathogen invasion and desiccation. In certain organs, epithelial cells are involved in absorption, secretion, and sensation. To remain as one cohesive unit, epithelial cells are tightly joined to each other into an underlying layer of connective tissue, known as the basement membrane. Epithelial cells are highly diverse and serve numerous functions depending on the identity of the organ in which they are found. In most organs, epithelial cells constitute the parenchyma. 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 Or the functional parts of the organ. For example, nephrons in the kidney are composed of epithelial cells, and hepatocytes in the liver, and acid-producing cells of the stomach are epithelial cells. Epithelial cells are often polarized, meaning that one side faces a lumen, the hollow inside of an organ or tube, or the outside world, while the other sides interact with underlying blood vessels and structural cells. For example, in the small intestine, one side of the cell will be involved in absorption of nutrients from the lumen, while the other side will be involved in releasing those nutrients into circulation for use in the rest of the body. We can classify different parts of epithelia according to the number of layers they have and the shape of their cells. Simple epithelia have one layer of cells. Stratified epithelia have multiple layers. Pseudostratified epithelia appear to have multiple layers due to differences in cell height, but are, in reality, only one layer. Turning to shape, cells may be classified as cuboidal, columnar, or squamous. What the hell? Cuboidal, columnar, or squamous. Squamous? Squamous. As their names imply, cuboidal cells are cube-shaped. Columnar cells are long and thin. Squamous cells are flat and scale-like. Pick your fighter. Pick your fighter. Pick your fighter. Connective tissue supports the body and provides a framework for the epithelial cells to carry out their functions. Whereas epithelial cells contribute to the parenchyma of an organ, connective tissues are the main contributors to the stroma, or support structure. Bone, cartilage, tendons, ligaments, adipose tissue, and blood are all examples of connective tissue. Most cells in connective tissues produce and secrete materials such as collagen and elastin, form the extracellular matrix. Kawaii. Classification and structure of prokaryotic cells. Prokaryotes are the simplest of all organisms and include all bacteria.
Prokaryotes do not contain any membrane-bound organelles, and their genetic material is organized into a single circular molecule of DNA, concentrated in an area of the cell called the nucleoid region. Despite the simplicity of prokaryotes, they are incredibly diverse. And knowledge of this diversity is essential for the study of medicine, because many prokaryotes can cause infection. In fact, choosing the appropriate antibiotic to fight an infection requires knowledge about the basic structure of the bacteria causing the infection. Well, prokaryotic domains. There are 3 overarching domains into which all life is classified: archaea, bacteria, and eukarya. 2 of these, archaea and bacteria, contain prokaryotes. Initially, archaea and bacteria were classified together into the kingdom of Monera. However, modern genetics and biochemical techniques have indicated that the differences in the evolutionary pathways between archaea and bacteria are at least as significant as between either of these domains and eukarya. Are you asleep yet? You know, there's 2 solutions to not being able to sleep. I got up from my bed. Why? Why? This should be like a podcast or something. You know, actually yesterday, I think I had one of... No, it was not the librarian's clips open. I would not be able to fall asleep to that. I think it's Fauna. I was watching her play some Pokemon, one of her Pokemon streams. And then, like, at some point, I forgot, I passed out, and then I woke up and it was like 4 a.m. And then, for some reason, she was on Getting Over It, and I was like, huh? How did I get here? Go to sleep, go to sleep. No, that's not the right one for this, right? Wouldn't it monitoring be better? Monitoring me? The stars align. Should I be doing monitoring? ねえ、私知ってるよ。どんな知ってるよ。ピクンピクンフルーツさ。でも、ただでもなんだわ。I think I totally botched the lyrics, but that's okay. Woohoo. Archaea, bacteria, eukarya. 3. Those 3. That's how you classify it. Life has to fall into one of those 3 categories. Bacteria, archaea, eukarya. Archaea are single-celled organisms that are visually similar to bacteria, but contain genes and several metabolic pathways. Metabolic pathways that are more similar to eukaryotes than bacteria. Historically, archaea were considered extremophiles, in that they were most commonly isolated from harsh environments with extremely high temperatures, high salinity, or no light. Salinity? Oh, I think that's like... Salinity is saline. I think the first word comes from saline. Like concentration? More recent research has demonstrated a greater variety of habitats for these organisms, including the human body. Archaea are notable for their ability to use alternative sources of energy. While some are photosynthetic, many are chemosynthetic and can generate from inorganic compounds, including sulfur and nitrogen-based compounds such as ammonia. Due to the similarities of this domain to eukaryotes, it is hypothesized that eukaryotes and the archaea share a common origin. Both eukaryotes and archaea start translation with methionine, contain similar RNA polymerases and associate their DNA with histones. However, archaea contain a singular circular chromosome, divide by binary fission or budding, and share a similar overall structure to bacteria. Interestingly, archaea are resistant to many antibiotics. All bacteria contain a cell membrane and cytoplasm, and some have flagella or fimbriae, similar to cilia. Because bacteria and eukaryotes often share analogous structures, it can be difficult to develop medicines that target only bacteria. However, in some cases, even seemingly similar structures have enough biochemical differences to allow the exclusive targeting of one kind of organism. For example, bacterial flagella and eukaryotic flagella are different enough that scientists are able to develop antibacterial vaccines that specifically target the bacterial flagellum. Also, many antibiotics target the bacterial ribosome, which is significantly smaller than the eukaryotic ribosome. They kind of look like pickles. Hairy pickles. Oh, hairy pickles. Bacteria perform essential functions for human beings, including the production of vitamin K in the intestine. Vitamin K is required for the production of the plasma proteins necessary for blood clotting. Newborn infants are not yet colonized by bacteria and cannot produce clotting factors, which put them at risk for hemorrhage. When babies are born, they are given an injection of vitamin K to aid in the production of clotting factors until they have been colonized with bacteria. That's crazy to think about, right? People think all bacteria are bad. No, actually, you're filled with millions of bacteria. There are approximately 5 times 10 to the 30 bacteria on Earth, outnumbering all of the plants and animals combined. As mentioned in the introduction to this chapter, bacteria outnumber human cells in the body by 10 to 1. The relationship between the human body and bacteria is complex. Some bacteria are mutualistic symbiotes, meaning that both humans and the bacteria benefit from the relationship. Examples include the bacteria in the human gut that produce vitamin K and biotin, vitamin B7, and which also prevent the overgrowth of harmful bacteria. Other bacteria are pathogens or parasites, meaning that they provide no advantage or benefit to the host, but rather cause disease. Pathogenic bacteria may live intracellularly or extracellularly. For example, Chlamydia trachomatis. Trachomatis? Whatever, chlamydia. A common sexually transmitted infection lives inside cells of the reproductive tract. Clostridium tetani, a cause of tetanus, lives outside the cells and produces toxins that enter the bloodstream. You know, if you don't like someone, a more posh way of telling someone that they should go die is that I hope you get chlamydia. I hope you get chlamydia. Or something like that. It's like, I hope. Did you know guys can get UTIs too? It's possible. It's rare, but you can also get UTIs. You know, I saw a clip today of a certain Eggman. He was like, where do I channel my inner road rage? And it was like, what did he say? You know, instead of like taking it all on the person, just imagine them like burning in hell, you know? But I think maybe that's a little bit too uncivilized. So a better way
think in both ways they're kind of freaked up. You just wish them. I hope you get chlamydia. I hope you get a UTI. I hope you get lots of dick cheese. You'd be so taken aback. You're
Classification of bacteria by shape. Classification of bacteria by shape provides scientists and pathologists, physicians who specialize in the infection and characterization of disease, common language to talk about bacteria. Provides a common language to talk about bacteria as well as to identify different shapes and species of bacteria. Most bacteria exist in one of the three shapes as shown in the figure below. Spherical bacteria, known as cocci. C-O-C-C-I. They're round balls. They're round balls, guys. You need a visual image. Just imagine a bunch of round balls. That's one form of bacteria. And then, common pathogen that's in that shape is Streptococcus pyogenes. Pyogenes. Have you heard of Streptococcus? Have you heard of strep throat infection? It's just when you have a bunch of these circular bacteria, these circular balls in your throat, when you infect the tissue area or cause the tissues in your throat to get inflamed or something, and then, yeah, that causes a lot of problems because you can't breathe properly. Everything else. Cocci, Staphylococcus aureus, bacilli, Mycobacterium tuberculosis, Spirilli. That sounds like pasta. It looks like pasta. Spirilli, Leptospira interrogans. Bacilli look like those do you guys know those Costco pretzels? Costco pretzels that are like in stick form? That's what bacilli look like. The giant box of pretzels. Or giant jar of pretzels. And then the spirilli kind of looks like, it low-key looks like a really long pretzel sticks. Yeah, instead of like the normal pretzel shape, it's like pretzel but like stick form. Cavatappi. Spirilli look like long cavatappi pasta. Very few pathogenic bacteria are spiral-shaped. The three most common are Treponema pallidum, the cause of syphilis, Borrelia burgdorferi, the cause of Lyme disease, Leptospira interrogans, the cause of Wales disease. This sounds like Italian brain rot. Borrelia burgdorferi. Something strawberry. My fucking Italian brain rot. Tung saur. Something cappuccino. I'm sorry. Sorry, all my Italians. Rod-shaped bacteria like Escherichia coli are known as bacilli. E. coli. E. coli are the things that look like pretzel sticks. Finally, spiral-shaped bacteria known as spirilli include the species Treponema pallidum, which cause syphilis. Why? Borrelia. Some bacteria require oxygen for survival, while others do not. Bacteria that require oxygen for metabolism are termed obligate anaerobes. Other bacteria that use fermentation or some other form of cellular metabolism that does not require oxygen are called anaerobes. Anna. Anne for no. Aerobe for air. There are different types of anaerobes. Anaerobes that cannot survive in an oxygen-containing environment are called obligate anaerobes. The presence of oxygen leads to the production of reactive oxygen-containing radicals in these species, which leads to cell death. Other bacteria can toggle between metabolic processes using oxygen for aerobic metabolism if it is present, and switching to anaerobic metabolism if it is not. These bacteria are called facultative anaerobes. Finally, aerotolerant anaerobes are unable to use oxygen for metabolism, but are not harmed by its presence in the environment. Oh, so there's aerotolerant? So it's like, it'll deal with it. It'll deal with oxygen if it's there, but it won't use it. Facultative anaerobes, the switches, obligates, which means that they cannot, absolutely not, no oxygen, and obligate aerobes. Oh, okay. One of the main differences between prokaryotes and eukaryotes is that prokaryotes lack a nucleus and a membrane-bound organelle. Prokaryotes are also single-celled organisms, meaning that each cell must be able to perform all the functions necessary for life on its own. However, prokaryotes may live in colonies with other cells and may signal these cells to share information about the environment. Prokaryotic cell structure. It's literally a sperm. This thing is a sperm, guys. The figure is just straight up a sperm. It's like there's that little rod with the little line dragging out from the end. Sperms are not prokaryotic cells, though, I don't think. Are they? Are they? Is sperm prokaryotic or eukaryotic? Sperm cells are eukaryotic. Oh my gosh. Whoa. Whoa. It's for science. It's for science. This is for science. I need. It can't just be all words. You have to be able to visualize. I'm helping you. I'm helping you to develop a very valuable skill. Can you see it? Unless you have aphantasia. Is it aphantasia? Yeah, aphantasia, where you can't see. You can't see images in your mind. Cell wall. Cell wall. Because prokaryotes do not form multicellular organisms, each bacterium is responsible for protecting itself from the environment. The cell wall forms the outer barrier of the cell. The next layer is the cell membrane, plasma membrane, which is composed of phospholipids similar to that of a eukaryote. Together, the cell wall and the cell membrane are known as the envelope. The cell wall both provides structure and controls the movement of solutes into and out of the bacterium. This allows the cell to maintain a concentration gradient relative to the environment. In the bacteria, there are two main types of cell wall: gram-positive and gram-negative. The type of cell wall is determined by the gram staining process, a crystal violet stain followed by a counter stain with a substance called safrarin. If the envelope absorbs the crystal violet stain, it will appear deep purple and the cell is said to be gram-positive. If the envelope does not absorb the crystal violet stain but absorbs the safrarin counter stain, then the cell will appear pink-red and is said to be gram-negative. Gram-positive cell walls consist of a thick peptidoglycan. A thick layer of peptidoglycan. peptidoglycan. I can't English today. Peptidoglycan. A polymeric substance made from amino acids and sugars. In addition to its structural and barrier functions, the cell wall may also aid a bacterial pathogen by providing protection from a host organism's immune system. In addition to peptidoglycan, the gram-positive cell wall also contains lipoteichoic acid. It is not clear what role this acid serves for the bacterium, but the human immune system may be activated by exposure to these chemicals. Gram-negative cell walls are very thin and also contain peptidoglycan, but in much smaller amounts. The peptidoglycan cell walls of these bacteria are adjacent to the cell membrane and are separated from the membrane by the periplasmic space. In addition to the cell wall and cell membrane, gram-negative bacteria also have an outer membrane containing polysaccharides and lipopolysaccharides. Interestingly, lipopolysaccharides are part of the gram-negative bacteria that triggers an immune response. In human beings, the inflammatory response to lipopolysaccharides is much stronger than the response to lipoteichoic acid. Wow. Bacteria contain a cell wall, the composition of which is different in gram-positive and gram-negative bacteria. Specific components of the cell wall can trigger an inflammatory response. Gram-positive bacteria have a peptidoglycan wall, and it's very thick, but it does not really have an outer membrane. It has a thin outer membrane, I think. Gram-negative cell walls, very thin. They do not have peptidoglycan, but then they have a thick outer membrane. The antibiotic penicillin targets the enzyme that catalyzes the cross-linking of peptidoglycan. Quiz: which cell? Which kind of bacteria involves peptidoglycan? Trick question, both of them. But gram-positive has more peptidoglycan. If a gram-positive cell cannot cross-link its cell wall, it no longer serves as an effective barrier. The bacterium then becomes susceptible to osmotic damage and lysis. Most bacteria have developed resistance mechanisms to penicillin, although a few bacteria, including Streptococcus pyogenes, which causes strep throat and some skin infections, and Treponema pallidum, causes syphilis, are still very sensitive to this antibiotic. Have you guys heard of multi-drug resistant bacteria? It's essentially what they were talking about in here, where sometimes bacteria, right, because they're essentially singular celled, what's going to happen to them is that they're going to randomly mutate. They're going to somehow obtain like a new gene or something that essentially allows them to, I guess, dodge whatever mechanism that drug uses to try to treat an infection. Like for penicillin works on an enzyme that essentially breaks down peptidoglycan, right? But if a bacteria becomes antibiotic resistant to penicillin, then that has to mean that they essentially have some alternative way of not having to utilize that enzyme anymore to build their peptidoglycan, or what they could do is they could somehow stop that antibiotic from binding to that enzyme that helps them keep their peptidoglycan, so then their cell wall can stay intact. That's crazy. It's crazy. That's how people get really bad infections. In the US, we have the worst cases of multi-drug resistant bacteria because we don't finish our meds. People tend to do this thing where if they start feeling better, right, they're like, oh, I don't have to take my meds anymore. They don't finish their antibiotics and stuff like that. But then, uh, what ends up happening, right, is even though you feel better, technically, some of those drug-resistant bacteria can still exist in your body, and you just don't know it. They could be dormant. They could be hiding out. They could be hiding out somewhere in you. And then, once you stop taking those antibiotics, they can essentially come out and start reproducing again. Scary? Yeah, I know. It's so crazy. So freaking crazy, man. Um, it gets to the point where, let's say your bacteria, because this has happened, your bacteria essentially becomes multi-drug resistant, right? So it, like, quite a bit of drugs have a hard time, like, working on the bacteria. If it gets to the point where even, because there's something called first line and second line drugs too. The first line drugs, those are typically the more weaker drugs. They have less side effects, they're less toxic. You want to use those first, right? Because you don't want people to have a severe reaction when they're already trying to deal with the side effects of an infection, right? You go for those, but then you have second line antibiotics too, and those are for very serious cases. You would think about those ones for typically, like, if maybe the patient becomes resistant to the first line drugs, but if the patient also becomes resistant to the second line drugs, that's when you would get a case known as like pan resistant. Pan coming from like panorama, like all encapsulating. You're essentially having an infection that cannot be treated with any medications. And in that case, you're kind of fucked, man. you're, you're actually so screwed. Because it's like no drugs can work on you. Your bacteria, or the bacteria causing the infection, essentially have so many freaking power ups or whatever, and like buffs that they can just dodge on your meds. Yeah. Mm-mm. I'm surprised they're talking so much about bacteria though, cause like this, I was expecting them to talk more about like our human cells, our eukaryotic cells. I didn't think they would go into microbiology that much. Just regular biology. Wow. Wow, 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 wow. Okay, we're continuing. Flagella. Flagella are long whip-like structures. Whip-like structures. I think that's the thing that's at the end of your sperm. Flagella. This is such a funny word. Flagella. That can be used for propulsion. Bacteria may have one, two, or many flagella. Depending on the species, flagella can be used to move towards food or away from toxins or immune cells. This ability of a cell to detect chemical stimuli. Oh. Chemical stimuli and move towards or away from them is called, oh my god, chemotaxis. Oh. Excuse me. Whoa. I'm gassy. The flagella are composed of a filament, basal body, and a hook. The filament, or what? Yeah, the filament is a hollow helical structure composed of flagellin. The basal body is a complex structure that anchors the flagellum to the cytoplasmic membrane, is also the motor of the flagellum, which rotates up to 300 Hz. The hook connects the filament and the basal body so that, as the basal body rotates, it exerts torque on the filament, which thereby spins and propels the bacterium forward. The overall structure of flagella is similar in both gram-positive and gram-negative bacteria, but there are slight differences due to the different physical structure and chemical composition of the envelope in gram-positive and gram-negative bacteria. Archaea also contain flagella, but the structure of their flagella is quite different from that of bacteria and is unlikely to be asked about. Huh. Oh my god. What the hell? Wait, this is so scary. Ugh. As mentioned earlier, prokaryotes concentrate DNA in the region of the cell known as the nucleoid region, which does not contain a nuclear envelope. Prokaryotic DNA is carried on a single circular chromosome, which can be found coiled around histone-like proteins in some bacteria. True histones are found in archaea. In addition, DNA acquired from external sources may also be carried on a smaller circular structure known as plasmids. Plasmids carry DNA that is not necessary for survival of the prokaryote and therefore is not considered part of the genome of the bacterium, but may confer an advantage such as antibiotic resistance. Prokaryotes lack several key organelles, such as mitochondria. Instead, the cell membrane is used for the electron transport chain and generation of ATP. Whoa. Prokaryotes do contain a primitive cytoskeleton, but is not nearly as complex as the one found in eukaryotes. Prokaryotes also contain ribosomes, but this ribosome is a different size from what is found in eukaryotes. Prokaryotic ribosomes contain 30S and 50S subunits, whereas eukaryotic ribosomes contain 40S and 60S subunits. The fact that prokaryotes and eukaryotes have different sized ribosomes imply that they carry out protein synthesis in slightly different ways. These differences are highlighted in blah, 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 blah. The differences also allow us to target bacterial ribosomes with a number of antibiotics, including tetracyclines, aminoglycosides, and macrolides, while leaving the eukaryotic ribosome more or less unaffected. Ow! Owie! I just gave myself a paper cut. That! You die! That. Books are dangerous. This is fucking Fahrenheit. Oh, Fahrenheit 450. 459? Am I botching the name of the book? Man. Yeah, books are dangerous sounds like some Fahrenheit 459 shit though, man. As we have seen, prokaryotic cells differ from eukaryotic cells both structurally and biochemically. Prokaryotic cells reproduce via asexual reproduction in the form of binary fission. In addition, prokaryotes are capable of acquiring and using genetic material from outside the cell. Wow. Binary fission is a simple form of asexual reproduction seen in prokaryotes. The circular chromosome attaches to the cell wall and replicates while the cell continues to grow in size. Eventually, the plasma membrane and the cell wall begin to grow inward along the midline of the cell to produce two identical daughter cells. Because binary fission requires fewer events than mitosis, it can proceed more rapidly. In fact, some strains of E. coli can replicate every 20 minutes under ideal growth conditions. You think about reproducing every 20 minutes? That's crazy, right? 20 minutes. 20 minutes, man. What the hell? They got game. They got game for real. This single circular chromosome of a prokaryotic cell contains the information that is necessary for the cell to survive and reproduce. However, many bacteria also contain extra genomic material known as plasmids. Plasmids often carry genes that impart some benefit to the bacterium, such as antibiotic resistance, the mechanisms of which are shown in blah 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 blah. Plasmids may also carry additional virulence factors or traits that increase pathogenicity, such as toxin predictions that allow attachment to certain kinds of cells, or features that allow evasion of the host immune system. A subset of plasmids called episomes are capable of integrating into the genome of the bacteria. Wow. God. Woo! Woo! Oh, okay. It's like a little ring. You know how DNA is normally in the form of like a helical structure, and then it's coiled into these balls, these histone balls, and then they're packed into like those like how do you describe chromosomes? Chromosomes are like if you stuck two pieces of like macaroni together, but like with their round ends next to each other. Like instead of that, you just have like a ring. You have a ring, and then that entire thing is just whole chain of genes. Whole chain of genes. That's what bacteria have, prokaryotes. Bacterial genetic recombination helps increase bacterial diversity and thus permits evolution of a bacterial species over time. These recombination processes include transformation, conjugation, and transduction. Transformation results from the integration of foreign genetic material into the host genome. This foreign genetic material most frequently comes from other bacteria that, upon lysing, spill their contents into the vicinity of a bacterium capable of transformation. Many gram-negative rods are able to carry out this process. Why? Conjugation is the bacterial form of mating. We're talking about copulation, but for bacteria. It involves two cells forming a conjugation bridge between them that facilitates the transfer of genetic material. The transfer is unidirectional, from the donor male to the recipient female. Oh. The bridge is made from appendages called sex pili that are found on the donor male. To form the pilus, bacteria must contain plasmids known as sex factors that contain the necessary genes. The best studied sex factor is F, fertility factor in E. coli. Bacteria possessing this plasmid are termed F plus cells, and those without are called F minus cells. During conjugation, the F plus cells replicate its F factor and donates the copy of the F minus cell, converting it to an F plus cell. This enables the cell obtaining the new plasmid to then transfer copies to other cells. This method of genetic recombination allows for rapid acquisition of antibiotic resistance or virulence factors throughout a colony, because other plasmids can also be passed through the conjugation bridge. The process of conjugation is illustrated in blah blah. The only thing I remember about conjugation is, like, remember how I was talking about there's like gram-negative bacteria and there's gram-positive bacteria? So typically what ends up happening is, like, let's say like a bacteria doesn't really have like a gene within it, right? There's no way you could technically like mutate it to cause it to appear. Like, what are we talking about? Like, let's say a bacteria doesn't already have like a sort of enzyme or protein channel present. It can't just mutate and appear. anything that it already has can be, but in this case, since it doesn't have it, right, you can't change it. You can't change something you don't have already. But if you do something like conjugation, where you have a different species, let's say a gram-positive in this case, gram-positive, gram-negative. Your original one was gram-negative. If you do something like conjugation, you can take stuff from one different species and put it into your other one. So typically you hear cases between gram-negative and gram-positive cells, bacterial cells. There's some more fucked up shit they do to make your infections worse. There's also another way they dodge your drugs. They're sneaky. The sex factor is a plasmid, but through processes such as transformation, it can become integrated into the host genome. In this case, when conjugation occurs, the entire genome replicates because it now contains the sex factor. The donor cell will then attempt to transfer an entire copy of its genome into the recipient. However, the bridge usually breaks before the full DNA sequence can be moved. Cells that have undergone this change are referred to by the abbreviation HFR for high frequency of recombination. Transduction is the only genetic recombination process that requires a vector, a virus that carries genetic material from one bacterium to another. Viruses are obligate intracellular pathogens, which means they cannot reproduce outside of a host cell. Because of this, bacteriophages, viruses that infect bacteria, can accidentally incorporate a segment of host DNA during assembly. When the bacteriophage infects another bacterium, it can release this trapped DNA into the new host cell. This transferred DNA can then integrate into the genome, giving the new host additional genes. The process of transduction is shown in blah 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 blah. Wow. Transposons. Oh. Let's see. Transposons are genetic elements capable of inserting and removing themselves from the genome. This phenomenon is not limited to prokaryotes. It has been seen in eukaryotes as well. If a transposon is inserted within a coding region of a gene, that gene may be disrupted. As discussed previously, bacteria reproduce via binary fission. I think that's it. Is there another one? There's not. Bless you. Thank you. Sorry. I just want to make sure another one wasn't coming. As discussed previously, bacteria reproduce via binary fission. This implies that all the bacteria are exactly the same in a local colony. There means no mutations or genetic recombination, and that no bacteria are dividing faster than the others. Bacteria can be said to grow in a series of phases. In the new environment, bacteria first adapt to the new local conditions during the lag phase. As the bacteria adapt, the rate of division increases, causing an exponential increase in the number of bacteria in the colony during the exponential phase, also called the log phase. As the number of bacteria in the colony grows, resources are often reduced. The reduction of resources slows reproduction. The reduction of resources slows reproduction, and the stationary phase results. After the bacteria have exceeded the ability of the environment to support the number of bacteria, a death phase occurs, marking the depletion of resources. One of the biggest challenges a doctor faces is patient compliance with treatment, especially antibiotics. Here's what I was talking about. Many patients fail to complete an entire course of antibiotics, often discontinuing the treatment because they feel better. Unfortunately, this breeds antibiotic resistance by killing off the bacteria that are non-resistant and leaving behind bacteria that are more resistant. These resistant bacteria then reproduce, resulting in recurrence of the infection. Over time, this practice has led to bacteria that are resistant so they don't respond well to multiple antibiotics, making common infections more difficult to treat. Wow. My 500 IQ brain. At the beginning of this chapter, we discussed the cell theory and noted that viruses do not fit the definition of living things because they are acellular. Viruses may be as small as 20 nanometers or as large as 300 nanometers. For references, prokaryotes are 1 to 10 micrometers, and eukaryotes are about 10 times larger. Unlike eukaryotic cells, viruses lack organelles and a nucleus. What is going on? So I was playing League and this is... Oi! You slacking? You slacking? No, I'm just kidding. I am unfortunately not reading Yaoi, though I wish I was. I'm reading bio. we're on viruses right now. No, there's how to... Oh, were you talking about this? Yeah, I'm outside your door reading Yaoi. Reading Yaoi. Yeah. I should say though, OTR, you probably remember this, right? Yaoi is biology. It's not as fun though. Not as fun. Where's my Omegaverse, man? Where's the biology of the Omegaverse? In a world where men can suddenly reproduce. Can you correct me though? The four parts of the cell theory, right? Is that all living things are made up of cells. That's the first one. The second one is that I think all or these cells have to be reproduced. Or no, that's the last one. That's the last principle. I think the second one has to be like... Nandake. See, I forgot this. Maybe I should say out of order. Then the second one is that cells have to come from pre-existing cells. Third one is that DNA is transferred in the form... Or sorry, genetic material... Or genetic information is transferred in the form of DNA. And then the third one is that... What is the third one? See, I don't remember the third one. Shoot. Oh my gosh. Let's just check it again. Ah! I don't remember. What is it called? Oh! The cell is the most basic functional unit of life. There you go. Energy flow and metabolism must occur within the cells? Yeah. That makes sense though. Because if you have to utilize... I think that comes from the one where it's like... the cells can only arise from pre-existing cells. Because you know how, like, viruses, they hijack other... Like a host or something to be able to, like... perform out some of their stuff? Metabolism and energy flow? Like that? You have to hijack a host material? Yeah. Viruses are composed of genetic... Did you close League? Did you guys close League? I'm saying you guys, as a collective. Viruses are composed of genetic material, protein coat, and sometimes an envelope containing lipids. The genetic information may be circular or linear, single or double-stranded, and composed of either DNA or RNA. The protein coat is known as a capsid, which may be surrounded by an envelope composed of phospholipids and virus-specific proteins. I'm about to frickin' bring out Getting Over It right now. The envelope is very sensitive to heat, detergents, and desiccation. Thus, enveloped viruses are easier to kill. On the other hand, viruses that do not have an envelope are more resistant to sterilization and are likely to persist on surfaces for an extended period of time. Pray that your viruses have envelopes. Because viruses cannot reproduce independently, they are considered obligate intracellular parasites. Viruses must express and replicate genetic information within a host cell because they lack ribosomes to carry out protein synthesis. After hijacking a cell's machinery, a virus will replicate and produce viral progeny, such as virons. Such as called virons. I'm not playing League? Likely story, Jimshi. Likely story. Bacteriophages are viruses that specifically target bacteria. They do not actually enter bacteria. They simply inject their genetic material, leaving the remaining structures outside the infected cell. In addition to a capsid, bacteriophages contain a tail sheath and tail fibers, as shown in figure 1.15. The tail sheath can act like a syringe, injecting genetic material into a bacterium. Tail fibers help the bacteriophage recognize and connect to the correct host cell. Make viruses at work by using plasmids and capsid-coating packing vectors? That's so cool. That's so freaking cool. I just learned about that in my lab. I'm playing Slay the Spire? What is Slay the Spire? I've been hearing people talk about that, but I don't know what it is. Biogenomes come in a variety of shapes and sizes. Some are made of only a few genes, while others have several hundreds. In addition, biogenomes may be made of either single or double-stranded DNA or RNA. Single-stranded RNA viruses may be positive sense or negative sense. Positive sense implies that the genome may be directly translated to functional proteins by the ribosomes of the host cell, just like mRNA. Negative sense RNA viruses are a bit more complicated. Negative sense RNA acts as a template for the synthesis of a complementary strand, which can then be used as a template for protein synthesis. Negative sense RNA viruses must carry an RNA replicase in the viron to ensure that the complementary strand is synthesized. Retroviruses are enveloped single-stranded RNA viruses in the family Retroviridae. Usually, the viron contains two identical RNA molecules. These viruses carry an enzyme known as reverse transcriptase, which synthesizes DNA from a single-stranded RNA. The DNA then integrates into the host cell genome, where it is replicated and transcribed as if it was the host cell's own DNA. This is a clever mechanism because the integration of the genetic material into the host cell genome allows the cell to be infected indefinitely. The only way to remove the infection is to kill the infected cell. The human immunodeficiency virus, HIV, is a retrovirus that utilizes this life cycle, which is one of the characteristics that makes HIV so difficult to treat. Both retroviruses and transduction are under investigation as methods of gene therapy. It is theorized that retroviral and transduction methods can deliver functional versions of missing or altered genes so that the correct proteins can be synthesized and certain diseases can be treated. Card game is like a roguelike deck builder, very addicting. Deck builder? Deck builder. Is it like... Sorry, I hope this is not a stupid question. You collect new cards, yeah. Is it like TFT? Does TFT not use cards too? I know TFT is not like League, even though they're both from the same company, but I think, yeah. TFT is a deck builder, but it's not the same thing? Question mark? If you consider Idols and Champions as a card. I wouldn't call TFT a deck builder. We have a discourse. Discord. Whoa, fusion. Oh. Hmm. Oh, not bad. Yay. Okay. We only have... We only have 2 pages left? Thank you. Basically, it's like... Sorry. Basically, in Slay the Spire, you collect cards that your character can use as actions. To attack, heal, block, etc. Some are more powerful than others. It's like the better version of like FGO. It's not just like block, attack, or like something else, right? Slay the Spire. Wow. As we have already discussed, viruses must infect a host cell and use the host cell's machinery in order to reproduce. Here we discuss the viral life cycle in detail. Viruses can only infect a specific set of cells because they must bind to specific receptors on the host cell. Without proper receptors, a cell is essentially invincible to the virus. Once the virus binds to the correct receptor, the virus and the cell are brought into close enough proximity to permit additional interactions. Enveloped viruses fuse with the plasma membrane of a cell, allowing entry of the viron into the host cell. Sometimes a host cell may mistake a virus bound to the membrane as nutrients or other useful molecules and will actually bring the virus into the cytoplasm via endocytosis. As mentioned earlier, bacteriophages use tail fibers to anchor themselves to the cell membrane and then inject their genome into the host bacterium through the tail sheath. Some tail fibers even have enzymatic activity, allowing for both penetration of the cell wall and the formation of pores in the cell membrane. Depending on the virus, different portions of the viron will be inserted to host cell. Enveloped viruses such as HIV fuse with the membrane and enter the cell intact, whereas bacteriophages only insert their genetic material, leaving their capsids outside the host cell. HIV must bind to a receptor called CCR5 on white blood cells in order to infect them. Some people lack this receptor and are thus immune to HIV. There was recently a case in which an HIV-positive individual with leukemia received a bone marrow transplant from a donor that lacked CCR5. Not only was the leukemia cured, but the patient experienced remission of the HIV infection because the white blood cells from the newly acquired bone marrow were not susceptible to HIV infection. This fire out 2 came out recently and is all in the brief right now. Double cure. Yeah. Venom, the little spiders. Gross. Gross. After infection, translation of viral genetic material must occur in order for the virus to reproduce. This requires translocation of the genetic material to the correct location in the cell. Most DNA viruses must enter the nucleus in order to be transcribed to mRNA. The mRNA then goes into the cytoplasm, where it is translated into proteins. Genetic material from positive sense RNA viruses stay in the cytoplasm, where it is directly translated into protein by host cell ribosomes. Native sense RNA viruses require synthesis of a complementary RNA strand via RNA replicase, which can then be translated to protein to form proteins. DNA formed through reverse transcription in retroviruses also travels to the nucleus, where it can be integrated into the host genome. That's a difference. Negative sense RNA viruses require synthesis of a complementary RNA strand via RNA replicase, which is then transported into protein, so it's not directly integrated into the DNA. Using the ribosomes, tRNA, amino acids, and enzymes of the host cell, viral RNA is translated into protein. Many of these proteins are structural capsid proteins and allow for the creation of new virions in the cytoplasm in the host cell. Once the viral genome has been replicated, it can be packaged within the capsid. Note that the viral genome must be returned to its original form before packaging. For example, retrovirus must transcribe new copies of their single-stranded RNA from the DNA that entered the host genome. A single virus may create anywhere from hundreds to many thousands of new virions within a single host cell. Viral progeny may be released in multiple ways. First, the viral invasion may initiate cell death, results in spilling of the viral progeny. Second, the host cell may lyse as a result of being filled with extremely large numbers of virions. Lysis is actually a disadvantage for the virus because the virus can no longer use the cell to carry out its life cycle. Finally, a virus can leave the cell by fusing with its plasma membrane as shown in figure 1.17. Process known as extrusion. This process helps keep the host cell alive and thus allows for the continued use of the host cell by the virus. A virus in this state is said to be in the productive cycle. Depending on growth conditions in the specific virus, bacteriophages may enter a lytic or lysogenic life cycle. These two phases are similar to the lysis and productive cycle methods of progeny release discussed above. During the lytic cycle, the bacteriophage maximizes the use of the cell's machinery, little required for the survival of the host cell. Once the host is swollen with new virions, the cell lyses, and other bacteria can be infected. Viruses in the lytic phase are termed virulent. In the event that the virus does not lyse the bacterium, it may integrate into the host genome as a provirus or prophage, initiating the lysogenic cycle. In this case, the virus will be replicated as the bacterium reproduces because it is now part of the host genome. Provirus or prophage. Although the virus may remain integrated into the host genome indefinitely, environmental factors, radiation, light, or chemicals may cause the provirus to leave the genome and revert to a lytic cycle. As mentioned earlier, the provirus may extract bacterial genes as it leaves the genome, which allows transduction of genes from one bacterium to another. Although bacteriophages can kill their hosts, integration of the phage into the host genome may actually benefit the bacterium. Infection with one strain of phage generally makes the bacterium less susceptible to superinfection. Simultaneous infection with other phages. Because the provirus is relatively innocuous, this arrangement may confer an evolutionary advantage. The lytic and lysogenic cycles are contrasted. Prions and viroids are very small particles can cause disease under certain circumstances. Prions are infectious proteins and are therefore non-living things. They cause disease by triggering misfolding of other proteins, usually through the conversion of a protein from a alpha helical structure to a beta pleated sheet. This drastically reduces the solubility of the protein as well as the ability of the cell to degrade the protein. Eventually, proteins protein aggregates form, interfering with cell function. Prions are known to cause bovine spongiform blah blah. Bovine spongiform encephalopathy, mad cow disease, Creutzfeldt-Jakob disease, and familial fatal insomnia. Humans. Viroids are small pathogens consisting of a very short circular stranded RNA that infect plants. Viroids can bind to a large number of RNA sequences and can silence genes in the plant genome. This prevents synthesis of necessary proteins, resulting in metabolic disruption and structural damage to the cell. Viroids are classically thought of as plant pathogens. But a few examples of human viroids do exist, including hepatitis D, virus alone. HCV is innocuous. However, when co-infected with hepatitis B virus, HDV is able to exert its silencing effect on human hepatocytes. Whoa! It can also cause cancer. That's why we have hepatitis C vaccines. Mad cow disease cannot be cured. If you get it, it's RIP. Oh. I forgot how mad cow disease spread again. Didn't they try to test it by feeding a dead cow to a living cow? Was that it? But yay! Guys, that was chapter 1. That was 40 pages, guys. Am I crazy or what? Angry cows. Origins of life. Wow! Wait! Praise me now. I'm gonna eat my apple. Oh, cheap bar. Sorry. Lisa. Thank you. OTR, you have to sit through and rewatch the entire thing. Since you missed it. Many sections. That's your studying. Just put me on 2 times speed, man. Sorry, I hope I didn't scare you guys yesterday. I was debating whether or not I should unarchive that getting over it VOD regular speed. What the hell? No, but then it's like you already know this shit, so you just listen on to like 2 times speed. You know, I also read really slowly. So then like if you listen to me at 2x, that's like how I should normally be speaking. I don't know all this stuff. You don't? What? Well, maybe, to be fair, I took a microbiology class right before this. So this felt like a lot of review for me. I'm using everyday work. You will soon, Dr. OTR. Yeah, anyways, I was thinking about unarchiving. Yeah, unarchiving the Getting Over It VOD because I was listening to it and I was like, oh my fucking god. You can't call someone who failed to get in 4 times a doctor? I can't. And technically you didn't fail. Because you still kept applying anyways, right? Wink, 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 wink. You're only not a doctor once you stop trying to apply. So, Also, I heard you were allowed to take the MCAT. Was it 10 times? 10 times before you're not allowed to take it anymore. So, that's also the other exception. Only exception. Where you can't become a doctor. 7 times? Oh, 7 times. Me when I spread misinformation. Yeah, I thought it was so cringe though. Sorry. I'm so sorry. But also, you know what? I was like, I'm near the limit. How many times you have left, OTR? How many times you got left? Yeah, make this one count then. Make this one count. Get that 525 OTR. 2? 2 left? Make it count then. Make it count. Feel free to reschedule your date too. Get yourself that 525, bro. I was like, what the fuck, Usa? Damn, you're so sad. Why? Why so serious, Usa? Why so serious? Yeah. Kawaii no. Did I reschedule mine? No. I'm sticking with it. I told you I'm superstitious. I just can't tell you when it's happening. Yeah. I'm sorry. You know what though? I feel like I wanted to test my limits, but like in weird ways. I should be testing my limits by like seeing how long I can study for, you know? But I've had getting over it in my library since like 2 years ago, 3 years ago, and I never played it. So I was like, you know what? Tape your balls back on, man. Go play getting over it. And I beat the fucking part that I was stuck on for like an entire week. I'm testing my limits by giving myself 2 weeks to hit my LCAP. Gumba. Crank out the practice problems. Crank out the U world. Now, take your FL now. After we finish reviewing. I believe in you. This dummy, this single-celled organism staring at you right now is trying it. You can too. I believe in you. See stuff for maintenance. Oh god. Kids are crashing the freaking website by checking their scores now. I feel like you could probably find it online. Not that I'm encouraging it, but I think you probably could. Yeah, guys, I was just on the Anki, yeah. Hey, I thought you were the one that said Anki was not as effective as actually doing practice, though. Also, I have work tomorrow. Are you gonna sleep then? Oyasubi. Oyasubi dasai. Otsukaresama deshita. Nom 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 nom. Mogu mogu. Mogu mogu. Obviously, there's a reason why I keep my VODs up, though. I keep it so that you can watch it at your leisure. That you don't have to feel pressured. You don't have to feel pressured to be here. Yeah? Because you can't go out first. It's not the same. Why not? Why not? When it's fun to talk to you, I can literally talk to you anytime. Which, by the way, I'm so sorry. Where's my sorry counter? How many am I at? I think I'm at 60. I'm not gonna say that number. 67. Fuck! I literally just I would say the number 69. I'm at 69. Sorry right now. You are not taking it too laxed. I think I need to clarify that. It's not you that's the problem, it's me. Rokunana. Rokunana. I need a better joke than that, man. I need a better joke than that. Luna literally has more freaking game than me. Or not game. What if I say game? What the hell? She's like 5. She has more jokes in her book than me. I'm scared, man. I'm scared for when she reaches like an L's age because she literally might
She literally might outdo everyone on this internet. On the internet, she might outdo all the VTubers out here right now who are only. She's gonna outdo me, whose only freaking jokes are poop and fart jokes, and dick and ball. Hee hee, poo poo caca doo doo. He was raised by the Eggman himself. Oh my gosh! You know what my favorite thing was? The one where they were playing that animal game or something, and then Anna was like, "This octopus, blah 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 blah," and she's like, in his perfect like intonation too. It's a squid. +2, +2. Get ratioed. It was crazy. But yeah, it's not you who's being too lax. It's me who's too much of a overthinker. You have to remember that, like, I've been worrying. I have an insane amount of stress and Asianness. Whoa, who said that? What? I'm not Asian. I'm American. My pronouns are not he, him. It's USA. Dun 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 dun. Bleh. I need to slow down. I have an abnormal amount of anxiety and whatever else inside this freaking small ass body of mine. Wait, speaking of Chinese though, guys, go look for that freaking clip of Ludwig speaking Chinese. It's probably on Twitter. The one where he confidently told the old man while he was on his China trip with Michael after the old guy was like, "Hey, your Chinese is really good." He said, "I'm a pussy burger." On tip to tip. Yeah, it's so funny. It's so freaking funny. I will find it. I will find it. I'll post it. I'll post it after this. Or you know what? I'll post it right now. He literally, the guy was being so nice too. He was like, "Oh my god, don't worry, your Chinese is so good." And he was like, 我是鸡鸡包炸鸡包。我是炸鸡包 or something. I'm crying. I'm actually fucking crying. He went into the funeral service thinking it was a restaurant and asked for food. No, that would be my 13th reason. That would be my 13th reason. Oh my gosh. I watched the freaking Squeaks Ludwig Yowie. I understand, dude. I understand. But tell me why Squeaks is so baby girl though. Squeaks is literally so baby girl. He's like my favorite bottom of 2026 at the moment. I love him. So pookyful. I'm vibing. I'm living my best life. If I hear that, mum, muggle. Hey, I think you forgot. You left me on scene. Hey. Do you guys know what I'm talking about? It's that one image of Mark Zuckerberg where it's like he looks like he's like borderline in tears, as if he's not always. And he's like, "Hey, okay, you left me on scene. You left me on red. Hey, hey." Guys, in my defense, well, actually not in my defense, because I woke up at 3 p.m. I freaking, what did I do? I woke up at 3 p.m. Okay, I woke up at 3 p.m. and I was like, "Are you fucking kidding me, man?" It's like 7 p.m. for you guys. It's so Jover, right? But then I noticed I've been keeping tabs, guys. I've been keeping tabs. I've been keeping tabs on your Oshis, okay? I know what times they stream, because I also like them. And I was waiting for them to be done. And I was just thinking, what's up? Actually, Ava's been hunting for jobs, which I'm literally rooting for her. Like, you go, girl. It's so freaking hard trying to get hired. And it's like, she's going for dental jobs too. I can't imagine her competing with all the dental students trying to get, like, internship experience. I'm pretty sure she got it. No, but that was after, like, her freaking 5th try. I've just seen, like, every single day, I've seen her post a tweet that's like, "Guys, please pray for me. I've just applied for another job." For dental office worker? Oh. I thought she was trying to go for dental assistant. And I was like, "Girl, what the hell?" That's too much, man. You wanna be a spit sucker? Saliva sucker? Yeah. No, but then in my defense then, I was camping out. I was camping out. I was waiting. I was like, when are your Oshis gonna be done streaming, you know? And like, I forgot. They're super professional about this, you know. They were built. They have the endurance to go for hours and hours, and they're very fun to listen to, you know. So then I lost track of time, but then also I have mad performance anxiety, guys. Like, I cannot go for that long. I'm tapping out. I'm literally going to tap out. I try to keep my VODs short so that you can watch them. You have bottom anxiety? Yes. Yes, I am. She needs to have either worked at a dental clinic or shadowed 200 hours. She ain't gonna shadow 200 hours because she wants to get paid. Dude, that was me. I literally, you do not understand how crazy 200 hours is. If you want to rack up 200 hours in a month alone as a fucking VTuber okay, this was when I was on my little on and off break from streaming and stuff back then. I was kind of shadowing in a hospital. I took a morning shift and a night shift, and I did that for multiple days in a week. Five full work weeks. Yeah, but that's both night shifts and day shifts too, though, bro. If you want it within a month. So that's all I'm saying, bro. All I'm saying. You have to imagine the kind of fear that was running through my body when I was driving on the road at like 2 a.m. in the morning. I paid work. Yippee! Yippee! Yeah. I can't though, man. I can't. Poor girl. No, it was fun though. I saw some crazy ass cases. I got to see some freaking crazy ass cases, and low-key, I do love my night shifts though. I do love my night shifts. I saw my. I said I'm not gonna out him like that. I'm not gonna out him like that. But tell me about it, right? Like, there'll be people like jaywalking and stuff like that, and it's foggy outside too, so they look like demons, and it's just like, I can't. I'm not. I won't. I can't. You'll never catch me on the road again, bro. You'll never catch me again on the road. Have you guys ever thought about the fact that driving is literally just a Mario Kart simulator, though? Why am I slurring on my words? Have you guys thought about the fact that driving is like a Mario Kart simulator? Every time you see a pedestrian, that's plus 10 points. Plus an extra 10 points if it's a child. Thank you for coming to my TED Talk. I'd rather not be hit by turtle shells than don't. Hit other people with turtle shells. Ma, take away her license. Don't let the DMV catch me, bro. Don't let the DMV catch me. They hate to see me on the road. They hate to see me on the freaking road. That's why I haven't been on it. So I literally will not drive. I'm fucking scared of people. I'm scared of people. They're scary, man. I don't want to put my life into your hands. I don't want to die at your hands, man. Well, I mean, I'm already dead. I'm technically already dead, but... Wait, I can't say that. I can't say I have 9 lives. I'm not a cat. I can't say that. I could be. Meow. Meow meow. Meow. Do you guys remember? Do you guys remember what that means? Meow. Meow. I'm hungry. I'm hungry. Feed me. Give me food. Meow. Meow meow. You're hungry. Yay! Rabbits die from random heart attacks? If I do... Wait, you just reminded me. ちょっと待って。待って待って待って。ちょっと待って。ちょっと待って。ちょっと待ってください。Mom, mom, 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 bum, 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 ba, 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 da, 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 da. Why am I singing in the Hall of the Mountain King? Guys, I'm so freaking excited, but I'm also really upset. Are you kidding me? Are you kidding me? She released a candle? A freaking candle? Sorry, I said I would save my voice. Listen to how good this sounds. Transport yourself into a blissful state of relaxation with drowsy dreamer. Bright notes of lemon and citrus wake up your senses, while warm embrace of spiced vanilla and candied coconut create a comforting atmosphere. Subtle hints of sugar musk add a touch of sweetness to bring this comfortable aroma together. This candle is perfect for those looking to unwind after the hustle of daily life, while you drift into a creamy oasis. Scent profile: lemon, citrus,
creamy spiced vanilla, candied coconut, sugar musk. You know what that is? You know what that is, guys? That's the freaking Lush super milk. It literally Lush's super milk. You guys didn't know? It's this spray that went viral. It went viral on TikTok and everywhere, and it's still people's favorites. It's like the second scent I hear recommended most, aside from freaking what is it, like Philosophy fresh cream. It smells. She says based on that. I fucking knew it. I fucking knew it. I clocked it. I was reading the scents earlier, and I was like, oh my god, I want it. I want it so bad. I'm not too familiar with this stuff. Even if you're not, I think if this is your gateway into getting candles, I think you should get it. I think you should get it. I am a whore for candles. I can make bath bombs though. No, but candles, they're just so dainty. They're so cute, you know. Like this one, it has a really cute piece of art on it, and then also, you know, the fact that it smells really good too. And the Lush Super Milk is freaking like $45. You can get that, but for 10. And it can help calm you down after a long day of work. You know, you're going to get to smell like a cute girl. Your room is going to get to smell like a cute girl. And that, in proxy, is essentially like you having a cute girl in your room with you. I feel bad using them. That's why you buy two. You buy one to burn it, and you buy the other one to huff it like your life depends on it. My TLDR to say please. Please buy the candle and then tell me what you think about it so that like I can live vicariously through you, because I can't buy the candle. I can't buy the candle right now. I'm so sad. Did you see what the owner initially suggested the name to be? No? Did they suggest to name it Mouse Girl Sweat? This is the equivalent of Gamer Girl Bathwater. I was the suggestion was Pillow Princess? Oh, wait, I can see that. No, yeah, cause her new outfit was really Pillow Princess coded, wasn't it? Wait, what is it called right now? Wait, sorry, I'm a fraud. Yeah, like bedroom royalty? Huh? What? Wait, what did they name it? Pillow Princess is not bad. Drowsy Dreamer. I mean, Drowsy Dreamer is cute. Pillow Princess could also be cute too, though. That is not what Pillow Princess means? A different meaning? The girl does not move? What? Girl does not move. Please Google Pillow Princess. Wait, I'm scared. I'm so scared. I've been saying that she looks very Pillow Princess coded. I've been saying that. I've been telling that to people. And I've been thinking in my mind too. What does it mean? Pillow Princess. Pillow Princess is a slang term for a person, often in a lesbian or queer relationship, who refers to receives. What
No fucking way. No fucking way. No fucking way. Guys? I'm glad I didn't say it to her. But now I'm wondering why. Thinking about it. Thinking about why people didn't sound as enthusiastic. And I was like, this is so Pillow Princess coded. on a on another day of the more you know. The fuck? Is it just a label for fucking everything now? Everything? Are you freaking kidding me? Pillow Princess? How long has that been a term, man? How long has that been a freaking term? Isn't that reasonable though? Like, I thought it would have been based off of like pillow forts or some shit. Very long time. No, but then you have to get where I'm coming from though. It's like, you know, people like during sleepovers and stuff, they build pillow forts and then they build like castles and shit like that. Like, wouldn't it be reasonable to assume that's where Pillow Princess comes from? Queen of the bedroom? Royalty of the bedroom. I can't say it with a straight face anymore. I can't do this, man. You know, I think you killed my like. It's not my Yume fantasy because like I don't shit myself with her, but then it's like I had a dream some time ago yesterday where I think we were playing Fogs again. But I was like, oh my god, your outfit's so cute. Like, I love your pajamas. You look like such a pillow princess. Blah, blah, blah. We were just laughing and having a good time, you know? Now I'm thinking, if that scenario were to play out in real life, I would get fucking owned. I would get owned so bad, man. That would be so funny. Now I can't forget it, man. I can't say it. She stared at you wide-eyed. I wish I hadn't told you. I'm never. We not a word. Not a word out of your mouth. You guys, this is going to the freaking grave. None of you guys. We're not gonna speak of this. This is our secret. This is immunity necklace, okay? I have to make myself look smarter than I actually am. This might be freaking worse than me telling my roommate that I thought Critter SMP was Critter Simps. I was like gaslighting myself. I was like, this makes so much freaking sense. When like Alice and like all of her other friends were doing the Minecraft server thing, my friend was like, no, Usa. SMP stands for survival multiplayer. Where the fuck did Sims come from? And I was like, I don't know. You were the one that added critters in front of the thing. It just makes sense. My brain just automatically went to people who love fucking animals.
Critter SMP. 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 Love critters! You love critters! Around multiplayer? Yeah, but it was labeled Critter SMP, and all the girls playing were freaking critters. Or there's some kind of animal. So I was like, wow, a server filled with people who love animals. Critter simps! Because I drew SMP, yeah, but I didn't know what the fuck SMP meant. Survival multiplayer. This tanks it, man. This fucking tanks it. Oh my god. I'm not gonna go to sleep after. You ever wish to... so awake now. No! No! Go back to sleep! What's the day to beam? What the fuck? Stop! I you don't have people in your playlist. Do you ever wish that you could unread something? Or just forget about a memory? This shit is going... You're underestimating how many times I've said that she looks so Pillow Princess coded. And now I'm scared. I'm so scared that it hasn't reached her directly. Thankfully. Thank fucking God. But I'm thinking about all the people that think I'm fucking weird now. Say like, oh my god, you're so princess. She looks so princess. Pillow Princess coded. Wow. So kawaii desu. But like, what the fuck is wrong with you? You freak. Should I play a convention stream? I can't, man. I can't do that. Sister Mimi is gonna send me the hell for that. What's the letter? Beam! Forget! Why the fuck would you do that? Why would you? I should be taking that to the grave with me. Not you. Not you. Anybody but you. Please, 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 please. Please don't tell anyone. Please. Please, I still have to look cool, sexy, and popular. I need to preserve this last bit of ego and self-esteem I have in myself. Please, 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 pretty, please, please, please. Oh my god! Oh my god! Did it work? Did it work? Are you convinced? Did I convince you? Hey. Hey, Hey. Hey. You, you're not. Right? Right? Right?
Guys, I'm doing everything I can to preserve this fucking my last sense of self-dignity. Please. There's been too many instances of this, man. There's literally been too many instances of this. I can't fucking do this. This is gonna keep me up at night, bro. Oh my gosh. No, 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 no. Listen. Listen, okay? That's just endearing. Endearing that I called one of my friends. I told one of my friends that they look so fucking pillow princess coded. Go to sleep early so you will wake up 3 p.m. No, no, no. You don't understand, okay? You don't understand what this is doing for me. Because I'm like, my brain is like going into fucking overdrive right now. There's a specific instance where I was playing Fogs, right? This is legitimate. This is not a dream. Unless it was. I don't know. Maybe I'm totally making this shit up right now. There's this instance in Fogs that I think about. I said to Alice, I was like, you know, I haven't really read like in a long time and stuff like that. Like I don't really consume media that much anymore, which is true. Like I'm not gonna deny that. And then what is it? I was asking her for Rex and stuff, right? And she was like, "Oh, well, what have you read?" I think that's what she said. I don't remember. And I was like, "Oh, you know, my favorite works are by Jane Austen." And then it's just silence. It's just silence after that. Just cricket, 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 cricket. I think about that moment. And I was just like, why did the conversation just die after that? And then I realized that is the most fucking performative shit. She froze on you. I think it fucking took her aback that I didn't fucking name drop some yaoi shit, and I was just like, Jane Austen. What do you like reading? Oh, you know, like I love Jane Austen. I think her stuff is so good. Like I love Pride and Prejudice. I sounded like a fucking 15-year-old, man. That's something a fucking 15-year-old would say to you. Like, is my reading comprehension or my reading level at that of a fucking like freshman? High school freshman? I kid you not. You don't understand. It might not seem like a big deal, right? But that sometimes I will be so tired, like exhausted, or like I'll collapse onto my bed. I'm like, thank God, I'm finally home. I close my eyes, and that one freaking instance just appears in full color in one. What's the highest? 180p. It shows up in freaking 180p in my mind, and I'm just like, oh my God, dude, just just jump off a bridge right now. Just jump off a bridge. Jane Austen is the fucking feminist writer of whatever. peak performative male's favorite author. She's the author that always, or the writer that keeps getting mentioned whenever like people are trying to like make satire comedy of like performative males, and then what is it, like when, like for example in those skits they'll be like, so I'm like 6'1", by the way. I love Jane Austen. I love reading Jane Austen. Why? Because she writes a bunch of fucking romance and whatever with female protagonists who are supposed to be strong, independent, and unrelenting to men. Something like that. I'm speaking to the void right now. And I'm defending myself to no one. Like, I should have just tanked this. I should have just tanked this and taken this with me to the grave. But you know what? I need to air it out. I need you to understand how fucking
how much of an overthinker I am, and how embarrassed I get. How embarrassed I get. I I I freaking thought about it. I meant it with pure 100% genuineness, 100% purity. I like Jane Austen. And I genuinely do. I do like Jane Austen. I'm not a performative male. I'm so sorry if I made you think that. It's getting credit your past self. That's why me finding out that I've been fucking calling one of my friends Pillow Princess Conan is gonna make me curl myself. Thank you. Thank you for coming to my TED Talk. My name is Yami Usuf, YouTuber. I hope you have a nice night, nice day. I am going to go curl myself in Minecraft. How did you even get that term in the first place? Which one? Performative male, Jane Austen, or Pillow Princess? You gotta be more specific. Pillow Princess? I don't. I'll hear the word in passing. I'll hear the word in passing and I'll think, wow, that's kind of funny. That's kind of colorful. I want to take that word. It's like the same thing with like edging. Edging. Like, quit edging me on. It's like, actually, no, that's no more ammo against me. The TLDR though is like, no, not even TLDR. Example. Okay, let's take edging for example. Okay, if I hear someone say, "Quit edging me on," in my mind, I'm thinking, "Oh, that's just a very colorful way, more interesting way to say, stop stalling." Stop stalling, right? So, I will adopt that into my vocabulary with that context. Same thing with Pillow Princess. I think I've seen Pillow Princess, like, I don't know if it was like in a movie or like, I know Bao had an outfit that was titled Pillow Princess. That was just like, oh, it's like, cause they saw her in her pajamas too. That's like bedwear, right? Was that what she was naming herself after? People was really together. Is it crazy that I didn't even realize that's what she meant until we had this conversation? Even now, I was still thinking like, oh, this is her pajamas. I'm smoking so much copium right now, man. You don't even understand. Technically, it's 99 because one of my classmates was like, what is it? My female classmates were like, you want to play chicken right now? I bet you won't. I bet you're scared. And I was like, I'm not. I'm scared of nothing. Nothing can scare me. So we played chicken, and I didn't back down. So it's 99. Yeah. Never bribe me with a game of chicken, man. I won't back down. Never back down. Never what? Never give up.
Man, you guys, this is fucking terrible. This is fucking terrible, dude. Internet footprint. This is what people meant by internet footprint. I can't do this. I can't do this. I'm literally gonna. My face is fucking red right now. My face is so red right now. Oh my god. Oh my god. What are we even talking about? How did I go from my existential crisis to learning about what Pillow Princess means? Candles? Candles. Nothing can change my love for candles though. I mean what I said. I think you should get candles. Which also, by the way, when did she even say this, man? I was literally sitting through her freaking doggy disco doggy disco? Doggy disco? That was fucking awesome. That was so cute. This is today's stream? Oh, with the fish? With the fish? What's it called? Guys, please. If you feel any remorse, please buy that candle. Please get that candle. Please get the candle and review it for me. If you feel any remorse, please. Please. I feel like that's scary. I feel like it has the opposite effect. Please get the candles. Please get the candles. I could donate so you can buy the candles yourself. No! No! It defeats the point. It can't be me. It can't be me taking the money. It has to be you. It has to be you willingly making that purchase. It has to be you willingly making that purchase. And it's not a random spend on your part if I was the one who encouraged you to do it and told you that it was worth it. Okay. This VOD is getting too long. This VOD is getting way too long. Unfortunately. Thank you guys, I guess. I'm just kidding. Thank you so much for keeping me company. I had a lot of fun. I hope you had a lot of fun too. I'm sorry for keeping you up this late. I was trying to be considerate of you. I was trying to be considerate of you or whatever. Thank you. Ochi ucha. I don't know. Since April is coming up soon, there is something I need to do, something quite important I need to do. So I don't know. I don't know how frequent my streams are gonna be, but I will try my best. I will let you guys know. I'll try not to be a bad streamer. Stream without telling you guys again. Thank you. Ocha ucha. Good night. Don't be like me and sleep at 8 a.m. in the morning and wake up at 3 p.m. Mata ne. Bye bye. Wait, let me do it properly. Mwah. At work, so I gotta go early. Bye bye.